We begin with mitosis in the cell cycle. This begins with interphase. Interphase occurs in both mitosis and meiosis, and cells spend roughly 90% of their time in interphase. There are three phases within interphase: G1, S, and G2. In G1, the cell grows and the genetic material takes the form of chromatin. Note that it is not diagrammed. Organelles will still reproduce here. In S phase, the cell goes through DNA synthesis, where the genetic material takes the form of chromosomes consisting of two sister chromatids. In G2, more growth occurs, as well as the preparation for mitosis. Prophase is the first phase of mitosis and takes the longest out of the four phases. In prophase, the nuclear envelope as well as the nucleus break down. Centrosomes and centrioles only being found in animals, migrate to opposite poles of the cell. Spindle fibers also form. These are made up of microtubules. Metaphase is the second phase in mitosis. In metaphase, the chromosomes line up at the equator or middle of the cell. The spindle fibers attach to the centromere, and there is one spindle fiber for each of the sister chromatids. Anaphase is the third phase in mitosis. In anaphase, the chromosomes begin to get pulled apart by the spindle fibers. Telophase/cytokinesis is the last part of mitosis. This is the opposite of prophase. The chromosomes are now on the opposite poles of the cell, and they unwind into chromatin. Next, we move to meiosis 1. This also begins with interphase. This interphase is identical to the interphase in mitosis. After this interphase, the cell moves to prophase 1. In prophase 1, the nuclear envelope and the nucleus break apart. Early spindles begin to form, and the centrosomes and centrioles migrate to opposite poles of the cell. Crossing over also occurs. Crossing over is when homologous chromosomes exchange genetic information. It is illustrated through the change of colors in the gummy worms. The site of crossing over is called the chiasma. Next is metaphase 1. In metaphase 1, the homologous chromosomes move to the equator of the cell, and the spindle fibers attach to the centromeres. There is one spindle fiber for each sister chromatid. Anaphase 1 occurs next. In anaphase 1, the chromosomes split apart and migrate to opposite poles of the cell. Spindle fibers are still attached to the sister chromatids. The final part of meiosis 1 is telophase 1/cytokinesis. In telophase 1/cytokinesis, the spindle fibers break down and the nucleus reforms, but not always all the way. The chromosomes are on the opposite poles, so a cleavage furrow forms and the cytoplasm divides, leaving two haploid cells. After meiosis 1 comes meiosis 2. Meiosis 2 is very similar to mitosis. Between meiosis 1 and 2, interphase does not occur again. The first part of meiosis 2 is prophase 2. In prophase 2, the nuclear envelope and nucleus begin to break down again. The spindle fibers reform, and the centrosomes and centrioles migrate to opposite poles. Note that all of these are occurring in both cells. Next is metaphase 2. In metaphase 2, the chromosomes migrate to the equator of the cells, and the spindle fibers attach to the centromere. There is one spindle fiber for each chromatid. Anaphase 2 occurs next. In anaphase 2, the centromeres divide and the sister chromatids move to the opposite poles of the cell. Lastly, telophase 2 and cytokinesis occur. In telophase 2/cytokinesis, the spindle fibers break down and a nucleus and nuclear envelope form around the chromosomes. A cleavage furrow forms in each of the cells and the cytoplasm divides. This ends with a product of 4 haploid cells that are all genetically unique from the one parent cell. This concludes the cell cycle and meiosis in animal cells. Thank you.